Hi everyone, how's everybody today? Yeah, it's good to hear. And so welcome to today's lecture on chronic disease. And what I'm going to do with this lecture is cut it into two parts. And why? There's a lot of information, so I think it'd be best to do it over a 2-week period. And the first one will be based on tuberculosis, and we're going to use tuberculosis as our case study. And it's a very interesting disease that has plagued us for probably the last 5,000 years and around there. So it's a really interesting type of disease to study. And then we're going to get into neoplasia in the following lecture. And that too is rather interesting, and it's a slightly different lecture. It's actually a difficult one to give online because I'd rather have a lot of audience participation in that particular lecture, but it's hard to do that online, so it'll be slightly different than I'm used to. Anyways, we'll continue, but first I have a series of announcements to make. So when we're looking at chronic disease, there's no clear dividing line between acute and chronic inflammation except for time. And the issue is that etiological agent, what the nature of that specific disease is. If we're looking at acute inflammation, remember, if that initial injury is not eliminated by AI, which the goal there is to go in, get rid of that etiological agent as quickly as possible, and then repair resolution. That's the game there. And it does so by those exudation reactions, fluid exudation followed by cellular exudation. If that doesn't work, then usually it will go to chronic inflammation. And when we're looking again at AI, don't forget, it's the exudations or reactions. That's what we're looking for. This is different than chronic inflammation. Chronic inflammation is characterized by a proliferative response, where we're stimulating these cells to essentially proliferate. And that's the name of the game there. And as we go a little further in depth, you'll see how closely related it is to the immune system. So having a good immune background, which you received last time, is going to help you understand this lecture pretty good. And unfortunately, most crippling diseases are chronic inflammation rather than AI. And we're talking leprosy, rheumatoid arthritis, tuberculosis, which will be our case study, liver cirrhosis, etc. All of these are very, very chronic diseases. You know, when we're looking at chronic inflammation, it can actually follow AI. It doesn't have to, but it can. In fact, you may have repeated bouts of AI following, then you have this chronic disease trying to creep in to resolve the scenario. It can't resolve it. Then you go back to a repeating bout of AI. And we saw this with a gastric ulcer, where you can have these repeating bouts of chronic inflammation and AI at the same time. It's this tissue damage, and that's the issue right there. And basically, it becomes very persistent. And that's the name of the game with chronic inflammation. Everything is persistent. And we're going to see this in the case of tuberculosis very, very shortly. So the hallmark signs of chronic inflammation includes basically the infiltration of these monocytes, these mononuclear cells, and they include monocytes, of course, and specifically macrophages, lymphocytes, and plasma cells. So macrophages, which originate from monocytes, as well as dendritic cells too, but we're going to keep that, we're just going to call macrophages and dendritic cells the same thing again for this specific type of lecture. Lymphocytes, both T cells and B cells, and plasma cells, which you know come from B cells. B cells differentiate into plasma cells. So if you're looking at these cell types, automatically think immunology. Why? Because these are the immune cells, essential for the immune system. Not only that, but another hallmark includes the proliferation of fibroblasts and vascular elements. Well, what does this mean to you? We took this a little while ago. If you're thinking granulation tissue, you are correct. In fact, when we're looking at chronic inflammation, we have immunology and repair. And these two processes are happening basically at the same time, but without resolution. That's the whole problem. So when we're looking at fibroblasts coming into the scene, and we're looking at these vascular elements, think granulation tissue. Not always, and we'll see that with tuberculosis. It doesn't always involve granulation tissue, but nevertheless, this is one of the hallmarks of chronic inflammation. Thus, you're going to get increased scarring, or what we call fibrosis. And you'll see this quite a bit, especially nowadays with COVID happening. One of the major outcomes that we see with COVID is basically fibrosis within your lung, and that's a problem. That's where the virus gets deep into the lung because it's a lower respiratory virus, and it causes quite a bit of damage within your lungs. It goes for the septal cells like we talked about earlier. It can cause cell death. That's localized tissue injury, and your body responds to that with a local inflammatory reaction. And that's the issue, whole issue of getting your lungs full of fluid. Anyways, back to this. When we're looking at chronic inflammation, we're looking at the irreversible destruction of our cells. This basically the stroma as well as the parenchyma. And when we're looking at it, it's quite reversible, especially if it's mild. But if not, then it's going to go chronic, and then you're going to have scarring. And that's the whole problem. So this fibroblast will come into the area with the vascular elements, laying down collagen, which you know now as scar tissue. But collagen is not always scar tissue. Don't forget that. It's a very natural molecule that basically makes up connective tissue, one of our connecting tissues that essentially help hold the body together, essentially. We'll learn more about that in HB 302. So we see this relationship between chronic inflammation and repair. But the problem is that etiological agent is still present. It's very tough to get rid of it, as you'll see in a little while with tuberculosis. And so as a result, you get to this scenario where you have chronic inflammation trying to go to repair, chronic inflammation trying to go to repair, and this vicious, never-ending cycle happens. And you can see where your body gets so frustrated, and the term that's derived from that is basically what we call frustrated repair, because of this persistent irritant. If we remove that etiological agent, whatever it is, then CI will go to repair. It'll go through repair to completion. And ultimately, that's what we want to do. And so your body has a handful of these responses that it uses to deal with these etiological agents. And one of them is with tuberculosis. And we'll have a look at that one very shortly. And everything, all the requirements for healing and repair is present. We have our immunology cells that's going to take care of that etiological agent. And then as time progresses, we have fibroblasts coming in, budding capillaries, and all of this is taking place at the same time. But unfortunately, it does so without result, because you have that persistent etiological agent. Get rid of that agent, it will go to completion. But if that agent's still there, then we get this frustrated repair situation where we're going to have some repair done, tissue damage, some repair done, tissue damage. And you get this vicious cycle that's happening. And, ooh. So let's look at the scenario with tuberculosis as our case study. And this is probably one of the more fascinating studies out there. There's two that really grip me. One is tuberculosis and the other one is HIV. And we'll deal with that particular virus at the end of the term. And this story is actually pretty interesting too, but it's really not as interesting as the HIV one, to tell you the truth. Why? Because more work has been done quicker with the HIV than has with tuberculosis. And in fact, when we're looking at tuberculosis, this is probably the worst disease that has ever hit humans in our history. And I'm going to try and convince you of that. I'm going to show you some slides and some data, and we'll see what happens. So what is tuberculosis? Well, it is a respiratory disease. It's caused by the bacterium, Mycobacterium tuberculosis. Cases, thank goodness, are declining in Canada and the States, but pretty much it's pandemic in other parts of the world, and that's the unfortunate part. The pathogenesis, and we'll go through quite a bit of detail about this, but just to give you some hints, we have three major types of tuberculosis. One is called primary TB, and this is the result of the initial infection. And it usually gives the initial granuloma that's formed. We call that Goens granuloma, Goens foci. It goes by a number of different names as well. Secondary TB, which is a reestablishment of this active infection after a period of dormancy. So this bacterium can actually live within your lung for a long, long period of time. We're talking decades in terms of its dormancy period. And we'll have a look about that strategy later on. And then the third type is disseminated, which is not the one you want, because this is the one that goes and spreads throughout your body. And this one always will result to death. So in terms of diagnostics, treatments, and prevention, we can diagnose TB. It's pretty straightforward. We use what we call tuberculin skin test. Chest x-rays is another good one to use in order to identify people with it. And when you look at the chest x-ray, you'll see all these little dots. Basically, they're pretty small. They can be up to about a few millimeters in terms of their size. And those are the granulomas that show up on the x-ray. Treatment is kind of brutal. And basically, common antimicrobials are pretty much ineffective. So you're going to use a combination therapy for quite a number of months. I had a colleague, and she was not diagnosed with tuberculosis, but she came in contact with that specific bacterium, and she went for the test. It showed up negative, but the doctor said, well, let's not take any chances. It seems that you were exposed, so let's deal with it now. And basically, she had to take 2 very powerful antibiotics for 6 months just to make sure everything was okay. And so this is for a person who didn't have it at all, but they wanted to be certain that it wasn't there at all. And we'll see what the effective dose is for this specific bacterium, and you'll understand why they decided to go this route, and a couple other features as to why they went this route too. Prevention. There is a vaccine that's available for it, and it's usually given to places where TB is quite common. But the problem with the vaccine is that it may not be totally effective. So there's question about the efficacy of this vaccination procedure. It seems there has been a number of trials and everything else, and one of the major trials that was done in the UK found a 70% efficacy, which is quite high, which is quite good. But for some reason, the closer you go to the equator, the efficacy drops, and no one really understands why that's happening. And so some of these developing countries which live near the equator, you can see that this could be an issue. And as a result, the search is on for modifications of this vaccine in order to get a better vaccination potential for this bacterium. So I'm showing you here where we have high incidences of basically tuberculosis outbreaks. And so you're looking for countries that are in this range here, even here. Okay? So North America, we're pretty good. Most of Europe is pretty good. Greenland is a hot spot. Some places in Africa, specifically South Africa, as well as up through the Congo area, and we're talking West Africa as well, Northwest Africa. And these are the places where you want that vaccination to take place. And the equator goes through and around this area here. So the efficacy hopefully would be better down in South Africa than it would be up into places like Cameroon and things like that. So why am I showing you a growth curve of the human population? Well, it's for a very particular reason. And years ago, I found a website where they gave out Nobel Prizes. And if you click on the link here, it'll take you to the Nobel laureate web page, specifically for Robert Koch, who did a lot of initial work on tuberculosis. And I told you his story about how he took his entire lab down to this medical stage because he was giving one of his lectures, and this was not his Nobel Prize lecture, but it was a lecture way before that, where he showed the entire audience his findings that have to do with tuberculosis. And, at the end of the talk, there was no applause, complete and utter silence. And people were in shock and horror of what his findings were. And when we're looking at the epidemiological evidence, and we guesstimate how many people have died from this disease, they estimated that between the period of 1700 and 1900, this disease has killed 1 billion people. And I'm going, you got to be kidding me. Look at the population at 1900 to begin with. We're probably somewhere around 1.5 billion people. And up to this point, just between 1700 and 1900, it's eliminating 1 billion people from that equation. So when we're guesstimating the population at the time, 1.5 billion, how do you fit that stat on a growth curve like this? And throughout history, we've had many epidemics. We've had the plague way down in here and up in here. You see little blips about how many people they've actually killed, those specific diseases. But when it comes to TB, you can't actually fit that data on here. It's incredibly difficult and challenging. So 1 billion people have perished because of this disease just between this 200 years. Unbelievable. Anyways, you can see all of a sudden we started growing. And what happened in this time and era, right around in here? You got it. Antibiotics, discovery of antibiotics, and increase in public health in terms of their procedures and how we deal with these types of viruses and bacteria. It continues on, but it's just an amazing fact. I just want to show it to you. Now, go to the website. They took away those stats for some reason because they redid their website, but the website's still pretty good in terms of the story about Robert Koch and everything else. So I encourage you to go. So let's back to the story. Let's talk about why this bacterium is so hardy and difficult to deal with. And one of the major reasons is because of its cell wall. And that's the problem. It's got a very waxy cell wall, very resistant to many, many features within our bodies, especially with water, and we'll deal with the other ones in a minute. It's called mycolic acid, and it really does have a number of unique characteristics. Because of the mycolic acid and another substance we'll get into in a few minutes, it really allows this bacterium to become what we call slow growing. And the cell cycle is very, very long for a bacterium. Basically, there's a number of different estimates out there, but 72 hours is a pretty good number for its cell cycle. That's in the ideal growth conditions. In your body, it may take longer for that to happen. So when we're looking at how this cell divides, because it takes a long time to synthesize the mycolic acid, it accounts for its slow growth potential. Not only does mycolic acid give rise to slow growth, but it also protects the cell from lysis, especially after phagocytosis. So this is one of the strategies it has to essentially deal with the phagosome fusing with the lysosome. And sometimes that happens, sometimes that doesn't happen. And one of the reasons is because of mycolic acid and another compound we're going to deal with very, very shortly. So it does give good capacity for its intracellular growth. In other words, this is an intracellular parasite again. Very resistant to the gram stain because of the mycolic acid. So in fact, we don't use the gram stain at all to essentially diagnose this Mycobacterium tuberculosis. We use what we call an acid fast stain, that procedure. So it's very resistant to gram stain, very resistant to detergents, and many antimicrobial drugs, and that's a problem. As well, desiccation. So here we have a figure just showing you where the mycolic acid is. So let's start down here. You know what the bacterial cell wall looks like. We have our cytoplasmic membrane, and then we have our thick peptidoglycan layer. So in essence, if you're going to put it into a specific category, it's really looking very similar to a gram-positive bacterium. And the mycolic acids stick right into the peptidoglycan layer. And then we have something what we call cord factor, which is on top of that. And it turns out that this is one of those really virulent factors, if you would. And we'll see that bacteria that have this cord factor are very, very infectious, and those that don't have it really are not as infectious. So virulent strains have this cord factor. What is it? It's a glycolipid. And the reason why they call it cord factor is when you're looking at when this bacterium does divide, it actually remains attached to its sister cells end to end. And so it forms these cords of daughter cells. And we'll show you a picture of that very, very shortly. In addition, cord factor inhibits migration of neutrophils and is actually cytotoxic to them. So it can actually kill neutrophils. So if there is an acute inflammatory reaction that's going to take place initially, which does sometimes happen, then it goes unresolved. Why? Because it has methods of dealing with the neutrophils. And that's the issue there. In addition, cord factor prevents fusion of endosomes, which are the phagosomes, and the lysosomes. So essentially, it's degrading that membrane structure within the cell. And once you're in the cell and you're not fusing with the phagosome, then you're going to be able to multiply within the cell. You're evading some of the immune system, and basically it's going to be a little bit harder to essentially deal with that bacterium. But you do have intracellular cytosolic receptors that can do some damage to that bacterium. Okay, don't forget about those. Refer to that specific lecture. In addition, the cord factor seems to be involved in what we call granuloma formation. It can actually stimulate cytokines that favor granuloma formation. And that leads to a very interesting theory, and we'll deal with that theory in a little while. So this is an image showing you the Mycobacterium tuberculosis bacterium. And this one has cord factor. Why? Because you can actually see long rows, if you would, almost, of bacterium. So this is an individual bacterium right here, and this is the daughter of it, and then keeps going down and down and down. So you have these long strings or cords showing you the bacterium. So we have many, many, many bacterial cells all along here. We have 1, 2, 3, 4, 5. And it goes down and down. You can actually see individual cells right in here. Another one there, another one there, another one there, forming these cords. So that's where that name originates. So what kind of inflammation does tuberculosis cause? Well, in us, and other animals too, one of the better models, if you would, model system that's being used is zebrafish. And if you refer to some of the articles that are posted on Quercus, one of them actually uses zebrafish as its model system. And it goes into the rationale as to why that is. So have a read of that. It's actually a very interesting story about TB. So what I want to tell you before we get into this type of inflammation is not all etiological agents will cause this type of chronic inflammation. So you have to understand that. Look back and see what essentially chronic inflammation is characterized by. It's that proliferative response of those mononuclear cells. Specifically, we're dealing with the macrophages, lymphocytes, plasma cells. And so all those mononuclear cells, we stimulate those to divide, to proliferate. That's the main goal And then we hook in the repair crew, fibroblasts coming into play, granulation tissue. So we have all the budding capillaries. So that's traditionally the route of chronic inflammation. Not all chronic inflammation etiological agents will give rise to this, but this one does. And other types, the cousin to Mycobacterium, the one that causes leprosy, is in the same family as this bacterium. It does invoke this type of inflammation. Asbestos is another type of etiological agent that can invoke this type of inflammation within your lungs as well. So what is this? Well, this agent that we call Mycobacterium tuberculosis, it really has a specific and distinct pattern. And we refer to this as granulomatous inflammation. And the characteristic feature, if you would, is called the granuloma. Now, before I go any further again, I just want to emphasize the fact that the ending here, oma, normally refers to something within something in neoplasia. Okay? This has nothing to do with neoplasia. This has nothing to do with cancer at all. It's just somewhat of a poorly named type of inflammation. They should name it something completely different, unfortunately. So granuloma, nothing to do with a tumor. Well, technically, in a way, when we look at the word tumor, what does the word tumor mean? It means any localized swelling, which can be neoplastic in terms of when we're dealing with cancer, or any other localized swelling could indicate some sort of acute inflammatory reaction. And in this case, we're getting this nodular-like formation. Basically, it's a nodule. And they can be large enough that you can actually see them. So we're talking up to a couple of millimeters in terms of their size. So you can actually see this on a X-ray. And so that's why they named it granuloma. It's a localized swelling within this chronic inflammation. So it's keeping with that tune. But some people do confuse it with some sort of benign type of tumor, and it's nothing to do with that. Okay? I want to be perfectly clear about that. Good. So what are granulomas? Well, they're small. We're talking 0.5 to a couple of millimeters in terms of its size and diameter. It's really a collection of modified macrophages. And they're modified in a very special way. They receive signals, cytokines, to basically to change their role a bit. And they become what we call epithelioid cells. Why? They call them epithelioid cells because their actual shape changes. It goes from, these big round structures to very squamous-like, almost very flat like a pancake almost, but they're a little bit thicker than that. And they take a more secretory role rather than phagocytotic in terms of the role that they usually do. And so we're talking secretion of mainly cytokines to attract other inflammatory cells, and specifically immune cells that are coming into play, specifically lymphocytes. And so a granuloma, by definition, is a collection of these modified macrophages, and then we see a rim surrounding this set of cells via the lymphocytes. And it gives it that very nodular appearance. And when we're looking at this, you can actually tell the color change between the different types of cells, which makes it very easy to distinguish what's going on in the granuloma. And we'll talk about that, and we'll show you pictures of that very shortly. In addition, as time goes on, other cytokines secreted by these epithelioid cells, and as well as lymphocytes too, will attract other cell types. Specifically, we're talking dendritic cells, we're talking more macrophages, and we're talking more of the immunology cells, as well as fibroblasts. And now we're getting into and trying to get to that repair crew into the scene, where we're getting mainly fibroblasts with just a few more of the capillaries. So we're not talking a lot of capillaries. It's mainly just the fibroblasts. And what do they do best? You got it. They lay down collagen. And we're talking fibrotic scar tissue that's being deposited. And basically, you want to try and encapsulate this bacterium within this granuloma structure. And we'll show you that very shortly. In addition, other cell types are going to be brought in. Neutrophils will be brought in. We have plasma cells coming in to try and secrete as many antibodies as it possibly can. And all of this is to deal with that persistent etiological agent. So when we're looking at what has to happen, first is you have to actually breathe in this bacterium via the respiratory droplets. This seems to be a very common theme, respiratory droplets. We're seeing this with COVID-19 as well as the flu virus and other viruses that cause respiratory infections. So this is a very common theme that we're dealing with, respiratory droplets. I find this kind of interesting. When we're looking at the minimum effective dose of this bacterium, we're talking just a handful. 10 bacterium is the minimum that's needed to essentially cause an infection. That's incredible. So this means that if you're in close proximity to someone who is active TB and they start sneezing and coughing, etc., and you get those respiratory droplets, the chances are pretty good that if you breathe these in, you will contract this bacterium. And as the bacterium gets deep, deep into your lung, it uses its pili. It's an adhesive pili in order to attach to our cells, specifically laminin. And what happens is we start to see the macrophages coming, usually they're first on the scene, and then it starts to essentially attract all the other cell types, specifically the helper T cells, to the scene. And then, so this is sort of like a modified type 4 hypersensitivity reaction that's taking place here. So have a review of that and see how that relates to tuberculosis. And these helper T cells, they start secreting a ton of factors. And they're what we call pro-inflammatory cytokines. And these are the most important ones. There is a whole list of cytokines that these things produce. But the important ones seem to be tumor necrosis factor alpha, gamma interferon, and interleukin 12. Now, interleukin 12 really does attract more T helper cells, specifically Th1 cells. And these factors are important in terms of the formation of the granuloma. They seem to be, if you don't use these factors, they essentially, you really don't get a granuloma forming at all. And to maintain the granuloma, tumor necrosis factor is essential for that formation, as well as to maintain it over time. If you get rid of tumor necrosis factor, you'll see that the granuloma essentially dissipates over time. So let's look at the development of this granuloma within basically a person's lung. So what are we dealing with first? Well, we have our initial bacterium coming in. You can see that it's on our alveolar surface. These are the cells. We call these pneumocytes. Specifically, this is pneumocyte 1. And this is the cell where essentially the oxygen goes through and the CO2 comes out. So these are very flat, squamous-like cells. And here they are, pictured in the cross-section right here, a very flat cell. And so we have the diffusion of those basically respiratory molecules in order for us to breathe and to essentially survive. So on that surface, we have the bacterium. You may get an initial PMN attack, and that would be basically unresolved. Macrophages will come in, engulf the bacterium, like we have here, and hopefully we don't get that fusion of the phagosome. We do get that fusion with the phagosome and the lysosome. We want that fusion to happen. If we do get that fusion, which we do, it's basically, as the story evolves, you see what kind of balancing act this is. And some people think that this is more or less wanted by the bacterium. And we'll talk about that theory a little bit later. But for now, we get the bacterium in the cell. Some will fuse with the phagosome/lysosome, and we'll get expression on the MHC2 molecules of this bacterial parts. The epitopes will be expressed on that. And we'll talk a bit more about that a little bit later too. If that happens, then we can start this whole cascade taking place where we get the attraction of more macrophages. And then as time develops, and we have the attraction here of more macrophages, engulfing the bacterium, and then as time progresses, we would see the attraction of all those other types of cells, specifically the lymphocytes. That's the name of the game at this point. So we have our initial tubercle formation right here. starting from a single, attracting more macrophages. And as you can see, we get this, what's called an incipient tubercle, and that's perfectly fine. This is the beginning of that. And as time progresses, we see different layers now appear. So here's all the macrophages. They're starting to become modified. So now they're getting more epithelioid in terms of their shape and what they do. So they become more secretory in terms of their role than phagocytotic. They're attracting lymphocytes to the area. So this next layer, if you would, is lymphocytes surrounding the initial tubercle that's been formed. And as time goes further, we start to see a layer of fibroblasts coming into play. So somewhere in between these two, we attract fibroblasts into the scene. And we'll see how that plays out very shortly. So as time progresses and basically the battle ensues within this area here, we start to see key features. One, cell death. So we're going to have dead bacterium, dead epithelioid cells, which are the macrophages. Not only that, but you're going to see live bacterium as well. And that's the problem with this case's necrotic area. It's mainly a very viscous looking type of necrosis. And don't forget, necrosis by definition is a pattern of tissue death. So within this area here, we're going to find many macrophages that are basically dead within this area. But if we were to do an acid-fast stain on this area, we would find that we have live bacteria too. Keep that in mind, okay, as we progress through the lecture. In addition, we have attracted all of this area here. All these lymphocytes are now coming into the scene. And again, you'll have live bacteria. And this very viscous type, almost like clumps, in many textbooks refer to this case's necrosis as cream cheese, where it's very, very thick in terms of what its structure is. Basically, it's a mass of debris, proteinous debris, that's scattered throughout this specific area, but we call a tubercle at this point. And this is a very oval, spherical type of structure. Anyways, so we have our case's necrosis, followed by a rim of lymphocytes, followed by a rim of fibroblasts. So what's happening as time progresses, and we're getting from early stage tubercle going into late stage tubercle. Late stage is really defined by these fibroblasts coming in and laying down collagen as much as they can. So we're trying to encase this bacterium within, basically, within scar tissue, within collagen. That's the name of the game here. And as time progresses even further, some of these areas of collagen are also actually going to calcify. So we get calcification happening within this outer area. What is calcification? Basically, you're trying to turn this outer rim, if you would, into stone, into rock, calcification, and hopefully sealing that bacterium in a tubercle forever. That's the name of the game. If you have good nutrition and you're taking those very strong antibiotics, that could be the likely outcome. And if your immune system's not compromised, that's probably what the outcome is going to be. But if you have poor nutrition, if you do have immune compromised system sometimes, if you're not taking antibiotics, then you can get what we call basically a rupturing of this tubercle. So this tubercle can rupture, and it can send this bacterium to other locations within your body. Specifically lung here, but it can travel to distant regions as well. And that's the problem with this type of bacterium. It can travel to other regions. And how does it do that? Well, as soon as you get bacteria being spread, you've got a very effective transportation system within you. It's the lymphatic system. And so we can actually scoop up the bacteria within the lymphatic system, because you're going to have quite a bit of inflammatory exudate happening within these regions too. And that's all being scooped up by the lymphatic system. And that's going to take it to the nearest lymph node, where it's going to try and, again, filter out and deal with these antigens. If it can deal with it, perfect. If it doesn't, then you get this scenario happening within those areas. And it can spread to other areas that have a lymphatic system. So when we're looking at this granuloma, the reason why I'm showing you this is to essentially show the initial stages and then how this granuloma starts forming, where we start attracting more and more of these basic cells that are involved with the immune system. And ultimately we get this, what we call the chronic granuloma, or late stage granuloma, where we're getting calcification hopefully happening within these areas. So the question is, who's benefiting from this granuloma structure? Because if we do an acid-fast stain on this, we find live bacteria. They're being encapsulated, sure, but they're still alive. Okay? Well, it turns out that when we're looking at the immunology of this, we have to define a new term to you, and that's immunodominant. And this is where we're looking at specifically the epitopes. And it turns out there's a range of them. So when the bacterium is engulfed within the cell, deal with the macrophage in this particular case, it breaks down the bacterium, and when it breaks it down, you have so many of these epitopes that can be placed on the MHC molecules. It's not even funny. There's just so many. And when we're looking at basically which one it chooses, there seems to be a priority, if you would. And that's the whole basis of immunodominance, where you're mounting an immune response to an epitope that really gives you a super strong result. And then you have the next one in the line, next one in the line after that, all the way down. And there seems to be a set handful that epitopes that we really mount a good immune response to. And this was first discovered in the flu virus and how your body responds to that. Well, it turns out it happens here as well. In fact, this pathogen has evolutionary conserved immunodominant areas within the CD4 and CD8 cells. And when we're looking at these epitopes, and you're looking at how these cells respond, it's producing epitopes that are part of this evolutionary conserved region. And it's part of the genetic makeup, the cell's genetic makeup, the bacterial cell genetic makeup. And it turns out that when you're actually producing antigens, it's actually the antigen dose that promotes this tissue necrosis, not really the bacillary dose. So we're looking at the response that's causing this antigen to be produced, and we have so much of this antigen that it actually promotes the tissue necrosis, which eventually leads to your body's response. So let me give you an example of this. And it's a specific protein that's being produced via the actual bacterium itself. It's what we call a virulence factor. And it's called ESAT6. This is one of the main ones. There's a number of different ones that they do produce, but ESAT6 is one that's studied quite a bit. ESAT stands for basically early secreted antigenic target. And what is it? It's a protein. It's one of these highly conserved proteins that's found within the bacterium. So our body mounts a very strong response to it. In fact, there are 40 highly conserved T cell epitopes, both for CD4 and CD8, from this one specific protein. 40, that's huge. And essentially, by immune dominance, there's a ranking of them and everything else, but this is one that is really used, not only used by our body, but it's also used, it's one of the main ones, one of the main epitopes that used within the vaccine that's being produced. So what does ESAT6 do? It is very constitutively expressed, this gene is expressed throughout the entire infectious process. The problem is ESAT6, which is basically a protein, and there's a huge story behind this, so I would highly encourage you to read one of the articles that is posted on Quarkus about this. It's very, very fascinating. That ESAT6 actually stimulates gamma interferon production. It stimulates tumor necrosis factor alpha production via macrophages. So mainly macrophages are doing this, plus T helper cells too. CD4 is a very good source of tumor necrosis factor alpha. And it also stimulates the production of more inflammatory, what we call pro-inflammatory cytokines. So this one molecule, ESAT6, does a lot of this. And so it's actually stimulating the formation of this granuloma. So why is it doing this? Is it to favor us or itself in terms of the bacterium for its own survival? At this stage of the game, it's just a theory, but it's a very interesting theory, because once it's encapsulated, you can actually see there's going to be live bacteria in here if you do that acid-fast stain. And if you're stimulating cells to secrete, especially tumor necrosis factor alpha, you're actually keeping that tubercle formed, because tumor necrosis alpha is essential to keep this granuloma formed. If you get rid of tumor necrosis factor alpha, basically this granuloma will, over time, be removed. So that's an interesting theory, where we have a highly conserved area on the bacterial genome that produces proteins that are extremely highly conserved that we mount an immune attack against. So if you look at it from both angles, from our perspective as well as the bacterium perspective, you can see where that does play a role. So we mount a response. It makes sense to mount a response against something that's highly conserved because it's there all the time. It's highly conserved. It's going to be, and this particular one protein is going to be expressed quite a bit. So our immune system is seeing this molecule, ESAT6. So we do mount a response against it. It makes sense. But the function of this protein, ESAT6, is to stimulate and keep the granuloma formed. Let me, let you think about that for a while. It's interesting. Who wins? For the last couple of years, I've been introducing this specific theory, and I still come to the same conclusion. Both win. We're at a stalemate by the sounds of it. So we are at an evolutionary stalemate. It sort of sounds that way. Why? Because once the bacteria is encased, and if your immune system's good, and you have good antibiotics, and you can actually keep that granuloma for an extremely long period of time. If you catch it at a young age, you can go through life basically like this. But there could be other factors that are played in the future where your immune system somehow becomes compromised, poor nutrition, and this granuloma may disseminate and spread the bacterium further. But the bacterium will still be alive. And that's the whole point. So at this point, I'm still saying it is a stalemate. Very interesting theory. So let's look at some images now, just to give you a context of how this bacterium basically, and what it does to you over time. So here we have a normal lung. And you can see the color is very typical of normal lung. And then we have one that's been highly infected with tuberculosis. Down in here is the initial contact. In terms of what we're looking at is we're just getting some of the bacterium down in here as well as here, but up in this part of the lung, the upper lung, you can see that basically it's disseminated basically within this area here. Huge areas of your lung has been replaced by casus necrosis / scar tissue, Over here as well, very nodular in terms of its appearance compared to down in here. So what's happening in here, especially up in this area here? Well, one of the major features is when we're looking at the amount of tissue that's damaged, it can actually cause what we call cavitations, which basically means that you're getting huge cavities in your lung. All of this was normal lung tissue, but now we have these huge areas where you're devoid of tissue completely. It's a big hole that's found there now. And all of this will eventually become this because your tissue is basically being dissolved, and that's really unfortunate. Normal lung on this side, you can see normal vessels and all of the nice little rosy areas here. All the little alveoli, you can't really see them very good, but you have your little tubes and everything else going down to smaller and smaller bronchioles. You can see you're still dealing with probably a segmental bronchus right there, because you can see cartilage in the outline, and then when you get to the holes where you don't have any cartilage, we're dealing with bronchioles at this particular stage, and these will all lead down into the alveoli down in here. So that's a normal view. And again, this is a closer view of a lung that's basically completely overrun with tuberculosis. And we're dealing with, again, cavitation, these big cavities that are forming within the lungs, here, and massive amounts of caseous necrotic areas, all of this area in here. So there's very little room for you to breathe. And you can see how these people will just suffocate over time. Very nasty type of disease. Now let's go down to the histology level. You've seen this image before. And what we're dealing with here is your normal lung. So here's a bronchiole, terminal bronchiole leading into the respiratory bronchiole, leading into the alveolar duct, because you have lots and lots of alveoli present, down into the alveolar sac here. So this is where respiration all takes place, all throughout here. So let's look at the tubercle and how we identify what they look like. So this is a very nasty case. We call this tuberculosis bronchial pneumonia. And this is where you have a huge chronic inflammatory response within your lung. And this basically were alveoli. You can see that they definitely do not look normal at all. But let's draw your attention to the tubercle. So this is a genuine structure that you can actually see. And let me show you some of the highlights. So we have our caseous necrotic area. This was the place where we have the epithelioid macrophages found. And then as time progresses, we have this big battle going on between the bacterium and the macrophages. We have huge area of necrosis formed because of that. This whole area is now caseous necrosis, which is indicative of tuberculosis. So this really, truly does look like a cream cheese type of substance that we're dealing with here. But if we did an acid-fast stain, you would actually see live bacteria in this area too. Now, as time progresses, the macrophages would attract lymphocytes. So if you can look out and you see this dark purpley color rim that surrounds this epithelioid cell region, those are the lymphocytes. So then you can actually see this rim that surrounds this area here. And then as time progresses even more, we start to attract fibroblasts out into this area. So this is pretty much a sort of I would say early mid-stage tubercle that essentially has spread. And it's definitely spread through different areas within your lung. This, you can see the outline of this wall here. This was a bronchiole, and this has disseminated into the bronchial area as well as other places within your lung, other alveoli. And this, you will basically find the formation of other tubercles within specific areas too. But I just want to show you a tubercle here, and we'll get more information about tubercles in a minute. And there are actually very special features found within these tubercles. And let's go and show you some right now. So in terms of other features, macrophages have an uncanny ability to fuse with other macrophages. And they do so, and they form what we call giant cells. And you can have up to 50 plus macrophages fusing together. And in the case of tuberculosis, they form a very specific type of giant cell, one that we call Langhans cells. And it gives a very characteristic pattern. And we'll show you that in a minute. The other type is what we call foreign body type giant cells. And if I was to describe them to you, the foreign body type has the nuclei of all these macrophages essentially as a uniform distribution throughout the entire cell. The Langhans version of these giant cells actually has a horseshoe-shaped arrangement of nuclei around the periphery of the cell itself. We actually see both types within tuberculosis. And Langhans giant cells are actually very indicative of tuberculosis. So it's a very easy guide, if you would. You can almost use it as a diagnostic feature because you rarely see them outside of tuberculosis. So what do they look like? We have both types depicted here. This is a beautiful one right here. This is the coalescence of many, many macrophages together, forming a giant cell. So this is quite big in terms of its size. And here we have the horseshoe-shaped arrangement of the nuclei going around the periphery of this specific cell. You can see this is a single nucleus, single nucleus, single nucleus, another one, another one. So they have this horseshoe-shaped arrangement. Here's another one, another horseshoe-shaped arrangement. Another one here, another one here, and there's many, many other ones too. If you look down here, you'll see an even distribution of nuclei throughout the cytoplasm. This is the foreign body type, so you actually see both within tuberculosis. Here's another one here too. But this is typical, and this is a Langhans giant cell. And this is a foreign body type of giant cell. Perfect. Now we're back to tubercles, and let me show you we have, by definition, an early stage tubercle. So what are we dealing with here? Well, we have our casus necrosis, this region here. This is all proteinous debris plus live bacteria, and you might find the live macrophage in there too. But nevertheless, this is all a pattern of tissue death, very indicative of tuberculosis, casus necrosis. Surrounding it, we have a rim of lymphocytes all around in here. So if I was going to draw you where all these epithelioid cells are, you can see that all this area that's not purpley in color, this would be where the epithelioid cells would be found. Okay? And the casus necrosis is growing out towards that, so it's going to encase as much as it can as it grows. And then surrounding, we have our lymphocytes. You can see all of these purple cells. This is the rim of lymphocytes. And then as time progresses, we'll start to see fibroblasts coming into the scene. So we have the initial stages of that happening here. But what I want to show you here are the giant cells. So here we have, and it's of the Langhans version. So you can see the peripheral location of the nuclei surrounding it. Here's a really good one here. You can see the periphery, the nuclei lining the periphery of the cell. And just a close-up of that, and you don't see too many nuclei within that region. But this is showing you casus necrosis, as well as the epithelioid cells that are within this area. As you go out, you can actually see the shapes of these cells. They're very squamous-like in terms of their shape, so they take a more secretory role. And as time progresses, we start to see the emergence of lymphocytes within the area. Now let's look at a late-stage tubercle. You can see they're very different from each other. In the middle again, let me outline the tubercle. Here's the tubercle here. We have our casus necrosis happening within this area. We have our epithelioid cells, followed by our lymphocytes within this area, and then we have our rim of fibroblasts coming into this area here. And fibroblasts do what fibroblasts do best. They lay down collagen. So this whole area is now surrounded with collagen. We're going for encasement here. And as time progresses, we're going to see calcification taking place. So calcification, you can actually see some of these nodular type of effects down in here, very solid. And you can see the odd one that's calcifying within these areas in here. This is what calcification would look like if you're looking at a late-stage tubercle. So this is very indicative of a late-stage tubercle. So if it doesn't spread, so you can see normal alveoli up here, if it doesn't spread, then it will be encased. But what we're trying to do here is essentially encase this granuloma in rock to keep it solidified. But it can still escape. The bacterium can still escape if you have immune compromise somehow, poor nutrition, etc., etc. Then it can break and then spread the bacterium to other sites. So really interesting theory about that in terms of, is this favoring the bacterium or is it favoring us? It seems to do both. So both have evolved this over a very, very long period of time. And again, if we did an acid fast stain, you would see live bacterium scattered throughout this tubercle. If it does spread, like you saw earlier, some of that's going to get scooped up into the lymphatic system, and this is what we're seeing here. This is the lymph node. And we have massive cases of necrosis happening within this region here. And you're attracting lymphocytes, because you had a ton of lymphocytes there to begin with too, and you're going to be attracting them in to do battle against the actual bacterium. So this is a very nasty case of tuberculosis that we see within a lymph node. So a summary of chronic inflammation. So we're dealing with a persistent vascular component as well. You don't forget, whenever you see that vascular component, think basically fibroblasts and budding capillaries. You may not have a large amount of budding capillaries. You may have other vascular elements that are present, okay, that gives rise to repair, and that's the whole purpose there. Persistent fluid exudation. So this is remnants of maybe the initial stages of acute inflammation, and this persists throughout. So you may have rebouts of AI, and you may see this persistent exudation. And probably the most important part of chronic inflammation is that these cells, lymphocytes, plasma cells, and macrophages, they're all being stimulated to proliferate. So we're attracting as many as we can, and we're getting them to proliferate. That's what we want to see. And as a result, you may see lymph nodes, nodules, basically regions where you would never normally see them. For example, in the kidney or something like that. You may see tubercles within the kidneys, depending on the etiological agent. In that particular case, it would be the Mycobacterium tuberculosis. So link CI and immunology. That's what we want you to do. I left this in. This is actually interesting. For the last few years, I've been collecting reports from what we call ProMed. It's a website that basically tracks infectious disease. And they come up with these word diagrams showing the world, of course, and seeing who's the major player in that particular year. So this is basically published in January of the following year. So this is published in January 2019, showing you what happened in 2018. And so we have 2018, 2019, and then I'll show you a surprise in a minute. So in 2018, the entire year, measles was the product basically everywhere within Europe. Huge, huge, huge. And you'll see that Ebola was a major one in Africa. It's actually not as much now, but there is currently an outbreak that's happening. We thought we had it under control within the Congo, and I think it's spreading within distant countries, adjacent countries now as well. Measles within South America, as well as Zika virus, that's actually predominant back in 2018, 2017. In North America, well, not a lot in terms of what's happening here. We didn't see a huge measles outbreak, but we did see a measles outbreak in the States. Quite a bit, especially in New York. And very interesting. So it doesn't give a huge, it gives a sort of big picture of what's going on at that particular time. And within 2019, so this was published in January of 2020, we see measles again, and a bit more polio, Ebola still, and Zika virus is taking over within this area here, hantavirus as well. And like I said, this is normally published from year to year to year. So the next one should have been January 2021. But something happened. You got it. So this was preliminary data they had. So this is, they're now publishing this in quarterly. So January to March 2020, one has taken over, huge. You can see within these areas here. And within the United States, quite a bit. And not so much in Canada, thank goodness. And the next one, April to June, even bigger. And so now we have countries that are really showing a huge effect. So the big countries are the India, South Africa especially, so this should be moved down in this area here, Brazil, and the United States. Those are the big hotspots in terms of COVID-19 right now. And this is showing why this is so dark and big. And they've reduced their numbers. I guess it would be more in towards Russia than anywhere else because they're showing huge case numbers right now. And it's huge. Here we have the report from July to September 2020. And you do see some changes, and they're quite a bit. You're looking at countries that are now being sort of engulfed by COVID. Brazil has been hit very hard now. As you can see, they're going through a rough stage, and we got to get those people vaccinated as soon as possible. Europe's still under it. The United States is still under it. In fact, they hit their milestone a few days ago in terms of they've hit 500,000 deaths, which is unbelievable. We're at current stages, we're at 2.2 million deaths, I believe, but I'll check that data and we'll go from there. It's taken a toll, and so vaccines are now being distributed. We have to get them out as soon as we possibly can. I'm hoping to have my vaccine sometime, with all the delays and everything else, probably sometime August, I would believe, in around there. So I'm home till then, yay! Anyways, so on to neoplasia next week. Beep, beep, beep. Attend lecture.